our reporter at GenomeWeb, and I'll be your moderator today. The title of today's webinar is How RNA Modifications Shape Cancer Biology, Epitranscriptome-Mediated Regulation in High-Risk Neuroblastoma, and is sponsored by Alida Bio. Our speakers today are Tanmoy Mondal, Associate Professor at the University of Gothenburg Sahlgrenska Academy, and Andrew Price, Vice President of R&D at Alida Bio. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears at the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Dr. Mondal.
A very warm welcome to this webinar, and thank you, Catherine, for introducing me to the audience. And I would like to start by thanking GenomeWeb for hosting this event, and also Alida Bio for supporting this event. I am Tanmoy. I am working as Associate Professor at Gothenburg University, and I'm also Swedish Childhood Cancer Foundation Senior Research Fellow. So today I'm going to talk about how RNA modification shape cancer biology. And our focus will be mainly high-risk neuroblastoma, which is a pediatric cancer. So before I go into the detail into today's presentation, I would like to give you a brief introduction, what we are going to cover today. So we will talk about the introduction, a little bit about the RNA modification, and then we'll also talk about what sort of methods we can use to detect RNA modification. And then a major part of the talk will be our research, how we have utilized RNA modification to understand the pediatric cancer neuroblastoma. And at the end, I will come back to the profiling methods which we are using to profile the RNA modification in neuroblastoma cells. So with that, coming back to what are the RNA modifications? These are chemical RNA modifications in the RNA molecules, also known as epitranscriptomic modification. And these modifications are could be quite diverse. And there are almost 50 modifications in the human cells. And out of those, some of these which you are seeing now in this screen. And as you can see that the RNA modifications could be driven by different writer enzymes. There might be different readers for those, and often sometimes they can be erased by other erasers. So it could be quite a dynamic regulation in the cells. And we will focus especially on one of the RNA modification called M6A, and I will come back to that a few minutes later. So why we will need to study RNA modification? So what we know that RNA modification can affect the different part of the RNA metabolism, such as RNA transport, translation, and degradation. We know that RNA modification can regulate the critical step of developmental biology, such as regulation of pluripotency factors. It can affect the several steps of oncogenic process, and that is something we will look today in the neuroblastoma perspective. And it can also control the immunogenicity in the cells, such as innate immunity. And it can also have an important role during infections. We and others have shown that RNA modification can have an important role during SARS-CoV-2 infection. And we are increasingly realizing the prognostic significance of RNA modification, which is not only in the cancer biology field, but in the other diseases as well. So to summarize, this demands that we need to be getting better to have a more reliable, fast, and cost-effective RNA modification method so that we can learn about the RNA modification function in the cells better. So with that, I will come back to the M6A RNA modification, which will be the major focus for today's presentation. M6A or N6-methyladenosine is one of the most prevalent internal modification in eukaryotic mRNA. And it is also not to forget that this is also present in the non-coding RNAs as well, which I will cover also during my presentation a bit in the neuroblastoma perspective. And this M6A modification is deposited in the mRNA by METTL3, METTL14, and WTOF complex. So these are the writer enzymes for this modification. And this can be also erased by some of the erasers. So that gives the dynamicity of the M6A modification in the cells. And this modification can be also recognized by reader proteins such as YTH family proteins. And once this reader recognizes this modification, they can then tell the cellular machinery for the RNA decay or RNA splicing function. So how this M6A is connected to the cancer field? And we have learned in recent past that M6A modification can be act as a prognostic markers, which we have seen recently in the breast cancer. It can regulate the oncogene function, such as the MYC oncogenes. It can also regulate the tumor suppressor function, such as p53 function. And we also know that it can regulate not only inside the tumor, but also the tumor microenvironment by regulating the homing of the immune cells. And recently, we also have realized that the M6A inhibition, which is by blocking AMT-TL3, the key enzyme of M6A modification, can be a therapeutic approach for cancers. And we will cover this part as well during our presentation in the neuroblastoma context. So what sort of method we can utilize to profile RNA modification or M6A RNA modification in the cells? So we have predominantly used a method called M6A RIPSEQ, where we have optimized this method in the lab to perform this assay using 3 to 5 microgram of RNA. And the method basically works in a quite a simplistic way. You take the RNA, you fragment it, and use the antibody to capture the fragmented RNA, and then you can sequence it to know what are the RNAs which are modified by the M6A. So it works, it's quite good, robust, and this is a technique which we have used in a majority of our study. But then one of the major limitation of this M6A RIPSEQ method is, as I mentioned, it requires 3 to 5 microgram of RNA. And that sometimes could be challenging if you want to profile the RNA modification in the clinically relevant samples, where the RNA amount will be quite limited. So towards the later part of my presentation, we will see that we have implemented now a new method called Epiplex. And Epiplex allow the RNA modification to perform in using only 200 to 300 nanogram of RNA. And not only that, the Epiplex-based method can also allow us to profile both M6A and inosine together simultaneously. I'm not going to go into the detail of this methodology because after my presentation, Andrew will take onto this method part, so he will be able to tell you much better way this detailing in this method, how the Epiplex works. But I'm going to come back to the Epiplex method towards the end of my presentation, and the other part of the studies where we have used the M6A RIPSEQ, I will also keep on mentioning them. So coming back to the disease, which we have focused, it's a neuroblastoma, pediatric cancer, as I mentioned. It's an extracranial solid tumor in children, and that cause around 15% of the pediatric cancer-related death. And usually the tumors are found in the adrenal gland, kidney, in spine. And the problem with the disease is the high-risk neuroblastoma, the patients, they often face high mortality rate. And even after quite an aggressive multimodal treatment, they do not respond to the therapy. So what we need to know that we need to understand the disease mechanism better so that we can treat this patient in a much effective way. And one of the hallmark of this disease is it's quite a heterogeneous disease, but as we will see during my first part of the presentation, the telomere maintenance mechanism can explain the part of this heterogeneity. And we will also see that how RNA modification can control the telomere maintenance mechanism in neuroblastoma. And then during the later part, we will see that neuroblastoma is a disease of differentiation, and this disease of differentiation is often associated with the amplification of an oncogene called MYCN. So we will also see how the RNA modification is connected to this MYCN oncogene function in the neuroblastoma, which will come back to into the second part of my presentation. So coming back to the telomeres maintenance mechanism in neuroblastoma, as I said, that it can explain partly the heterogeneity in the neuroblastoma tumors. So usually we see that the high-risk neuroblastoma tumors, they are often positive for telomeres maintenance mechanism. And telomeres maintenance mechanism can be governed by quite a few of the ways, but the one which we are interested they are all positive tumors or alternative lengthening of telomere positive tumors. And these tumors can contribute to the almost like 22% of the high-risk tumors. And in the relapsed cases, it might go as high as 47.5%. And unfortunately, these all positive neuroblastoma tumors often relapse and do not respond to the currently available therapy. So what are these all positive tumors? So all positive tumors are TERT negative. They are high risk tumors, and usually they contain very long telomere. And we do not understand the alt mechanism very well. But what we know that a long non-coding RNA is actually produced from the telomere ends in these cells. And this long non-coding RNA is named teraRNA, and it contains the telomeric repeats in many times in this RNA. So Roshan Bait, who is a senior postdoctoral research in my group, he wanted to sort of took this project and wanted to sort of know how the teraRNA function in the alt positive neuroblastoma. So what he did, the first experiment he did, he wanted to sort of know expression of this teraRNA in alt positive tumor versus alt negative tumors. And what he saw is a very clear trend that in the alt positive NB tumors, tera expression was quite high. So that immediately tells us that this must be doing some sort of important function in the alt positive neuroblastoma. And if you try to look where this teraRNA is located, which is marked here by these green dots, they are usually located in over the telomere, which is marked here by the red. And you can see very nice overlap of green and red signal. And if you stain the all positive cells with a immunostaining with the M6A antibody, you can see that the tera and M6A very nicely overlap as well. So that tells us that teraRNA could be M6A modified. And that is what exactly the clue which Roshan took forward. And he performed a M6A immunoprecipitation experiment. And what he could see that the teraRNA was very robustly enriched with the M6A modification in the all positive cells. And he did then series of experiments very critically to show that the M6A modification was quite enriched in the telomeric repeats of teraRNA molecules. But then we know from the literature that tera doesn't originate from the one telomere ends. It can come from the multiple telomere ends. And Akram Mendez, who was a postdoc and bioinformatician in my research group, he wanted to look where from the teraRNA originates in the neuroblastoma cells, which telomeres are sort of active for teraRNA. And he found that a group of telomeres are active in the neuroblastoma cells, which you see has a dark purple color. And what are the light purple color one, which are sort of negative of tera expression. And not only that, he found that several of those highly expressed tera expressing telomeres were also enriched with the M6A modification. And that you see by the dark black dots. And this is one of the representation of such chromosome ends, which is enriched with the M6A modification. So this is the data coming from the all positive NB tumors. And we also saw that the similar trend in the all positive NB cell lines as well. So this is the cell lines, which is XKNF1, which is the all positive NB cell lines. And you can see very nicely that M6A modification is quite heavily enriched in the telomere in these cells, but this enrichment is much lower in the alt negative cells. So overall, this data suggested that the M6A modification in the tera could have important function. But then we need to know, right, what sort of function that is. So that is exactly what we did. What we did, we actually removed or knocked down on the main M6A methyl transferase, which is METTL3 in these cells. And what we saw that, though in the control cells, you can see very nicely the tera foci, those foci were actually reduced or completely gone when you block the METTL3. And concomitantly, what we see that along with the tera loss, in these METTL3 knockdown cells, the telomeres are getting damaged as well. It accumulates the gamma H2X, which is a marker of telomere damage. So Rebecca, she was a postdoctoral researcher as well in my group, and expertized the animal experiment, took that clue, and she wanted to see that if this METTL3 knockdown has any effect in the neuroblastoma, all positive neuroblastoma cell growth. So what she did, she engrafted these cells into the mouse in a xenografted assay, both in the control cells and the METTL3 knockdown cells. And what she found that METTL3 knockdown leads to drastic reduction in the xenograft of all positive neuroblastoma. So we sort of concluded from this experiment that the MET-3 was required for all positive neuroblastoma growth. We also wanted to explore it a bit in the therapeutic way, and for that we use an inhibitor for the MET-3 called STM2457. And if you use this inhibitor in the cells, what we saw that MET-3 inhibition also leads to loss of terrafoci, as we saw in the MET-3 knockdown cells. So basically, it phenocopied MET-3 knockdown cells. And this terrafoci loss also concomitantly leads to increase in the gamma H2X accumulation in the all positive neuroblastoma cells only, where you see that in the SKNF1 cells. But if you consider the all negative cells such as SKNB2 and SKNS cells, the other neuroblastoma cells, this sort of accumulation was not there. So what we overall concluded that M6A RNA modification in the tera-RNA plays a critical role in the telomere maintenance in the all positive neuroblastoma. And we also sort of proposed that targeting M6A modification by MET-3 inhibition might provide a therapeutic option for all positive neuroblastoma, and which is important to highlight that a proper therapeutic strategy for all positive neuroblastoma is currently lacking. So with that, I will switch a gear a bit. I did tell you in the beginning that we are going to talk about the differentiation problem in the neuroblastoma, and that usually happens with the MYCN amplification. So now, in the next few slides, I'm going to talk about another high-risk neuroblastoma, which is usually associated with the MYCN oncogene amplification. And we often see that 50% of the high-risk tumors are associated with the MYCN oncogenes, and MYCN oncogene amplification is usually associated with the poor disease outcome, which you can also see nicely in this survival plot analysis. And histopathologically, if you see this tumor, you will always see the poorly undifferentiated phenotype in these tumors, and they are often categorized as an unfavorable type of high-risk tumors. And in this project, our main goal was to understand if RNA modification has any role in the controlling this MYCN function or MYCN oncogene function in the neuroblastoma. So if you go back into the disease, how this disease happens, we know from the literature that the trunk neural crest cells, which normally gives the sympathetic neurons, if they fails to differentiate, they results in the neuroblastoma tumor formation. And this failing of differentiation can be sort of driven by MYCN oncogene amplification or different site of chromosomal aberrations such as 17q gain and 11q gain. So when this sort of chromosomal aberration happens, the cells which is normally supposed to form sympathetic neurons now doesn't differentiate well and results in the neuroblastoma tumor formation. So our goal was in this project that if we can mimic this trunk neural crest differentiation in vitro, that will allow us to sort of model the disease well. And using that model, then we can explore the RNA modification function. So that is a project. It was taken by Ketan Thambare, who was a PhD student in my group. He defended a few months back. So what Ketan did, he took embryonic stem cells, and he driven those embryonic stem cells to trunk neural crest cells. And these cells can be actually further differentiated to sympathoadrenal progenitor cells, and then finally to the sympathetic neurons. And as you can see very nicely from this different and particular staining pattern, which are specific for different stages, this differentiation works fantastic. So when we have this differentiation strategy in our hand, Ketan wanted to look if the key M6A RNA modifying enzymes are sort of having differential expression during this differentiation process. And what Ketan observed that the key M6A modifying enzymes are usually having very high expression in the tNCC stage, but as the cells differentiate from tNCCs to SAP and SN, this expression of their expression goes down. So this tells us that probably they have an important function during this differentiation process. But what sort of regulation they have then in these cells? That is what exactly Ketan wanted to look. So what he did, he actually knocked down MET-3 in these cells. And once he knocked down these cells, and then he differentiated these cells to the sympathetic neurons. And what he found that if you knock down MET-3, we see that the differentiation was much better. And this can be measured by neurite length or even the positivity of the SN markers such as PRPH. But then we wanted to know that what are the reason why if you remove MET-TEL3, the cells differentiate better. So we performed RNA-seq analysis, and the RNA-seq analysis actually gave us quite a lot of genes. Among those, we saw that the HOX genes were changing. They were upregulated in the MET-TEL3 knockdown cells. And among those HOX genes, there was critical few HOX genes which are called posterior HOX genes, such as HOXA8 and HOXA9. They were upregulated. And we know from the literature that these are very important for the neural crest differentiation. And you can see here a qPCR verification of our gene expression analysis that in the MET-TEL3 knockdown cells, the HOX9 and HOXA8 and HOXA9 expression goes up. And not only that, M6A RIPSEQ profiling in these cells in the trunk neural crest suggests that the HOX8 and HOXA9 mRNA is very heavily M6A methylated as well. But this is the data which comes from in vitro, right? And you always want to know that, is it something related to the disease? And that is what exactly we wanted to look in. So we found that the HOXA9 expression is actually quite downregulated in the MYCN-amplified neuroblastoma in the RNA-seq data. And I have presented here HOXA9, but the situation is same for HOXA8 as well. And if you try to look the HOXA9 expression using an immunoblot, you can clearly see that the MYCN-amplified tumors have a reduced expression of HOXA9 compared to the non-MYCN-amplified neuroblastoma tumors. So this suggests that the HOXA9 expression is usually down. But are there M6A modified? We actually do see in the MYCN tumors, HOXA8 and HOXA9s are also robustly M6A modified as well. So far what we see that the MET-TEL3 has an important role in the neural crest cell differentiation. But if you could remember that couple of slides before, what I told, that we wanted to use this tNCC differentiation system to create a neuroblastoma model, MYCN-dependent neuroblastoma model. So what Ketan did, Ketan actually looked for MYCN expression and during this process as well. And what he found that the MYCN expression was very high in trunk neural crest cells, but as the cells differentiate, the MYCN expression is drastically diminished. So what he wanted to do, instead of, when the normally MYCN expression goes down, he wanted to keep the MYCN expression always on in this system. And that will somehow help us to mimic the neuroblastoma scenario, right? Because there we have a lot of MYCN expression which comes from MYCN oncogene amplification. So now if you look into the SAP stage, which normally you see that the MYCN expression goes down there, but when Ketan actually switch on MYCN, you see a lot of MYCN now present in these cells, and which is a control cell is minus dock cells, where the MYCN has not been switched on. And this MYCN expression can be maintained until the SN stage as well. Okay, so now we have able to achieve the sustained MYCN expression, but does that lead to any phenotype? And I think the phenotypic data looks quite nice. So these are the control cells. And if you do not switch on MYCN, these cells differentiate very well. They form beautiful sympathetic neurons. But the moment you switch on the MYCN in these cells, what you see that these cells doesn't differentiate. So looks like we could actually mimic what happened in the neuroblastoma situation, where you see undifferentiated cells because of MYCN oncogene amplification. And not only this differentiation phenotype we could see, we could also see that the critical trunk neural crest genes, such as HOXA8 and HOXA9, were also downregulated as well. So now you see, we see actually two different patterns, right? In the MET-TEL3 knockdown, we saw that differentiation was more. We saw that the HOXA8 and HOXA9 expression was upregulated, whereas in the MYCN overexpression scenario, we see the differentiation is perturbed, and then HOXA8 and HOXA9 expression is low. But that tells us, looks like these two proteins sort of crosstalk with each other. But do they really crosstalk? That is what exactly we wanted to test. And this is the data which shows that they actually really do crosstalk with each other. So we have performed a crow-immunoprecipitation experiment, which clearly suggests that they physically, MYCN and MET-TEL3 physically interact with each other. We also perform an alternative experiment, which is called proximity ligation assay, where if two proteins are close together, you will get a nice signal. And this signal is here in the green, which you see in the tNCC cells. And we could also reproduce that in a cells where normally lacks MYCN, but if you forcefully overexpress MYCN, there also you could see that MYCN and MET-TEL3 interact with each other. But they interact with each other. But do they co-localize in the genome? That is what we wanted to test. And we perform a chip-seq analysis with the MET-TEL3 and MYCN in the tNCC cells. And what we could see that MYCN and MET-TEL3 largely co-localized in many parts of the genome, and including, and this co-localization also happens in HOXA8 and HOXA9 locus as well. So what overall we think that MYCN and MET-TEL3 sort of interact with each other. MET-TEL3 was brought to the target sites by MYCN, and it can regulate the M6 methylation in many genes such as HOXA8 and HOXA9. Okay, so far we saw that the developmental aspects of this disease, right? But we sort of seen that the MYCN and MET-TEL3 connection can regulate the timing of HOX expression in this system. But does it really have a therapeutic implication? That is what exactly Ketan wanted to explore further. So these are the cells where if you switch on MYCN, they don't differentiate, which we saw in few slides before. But in the same cells, now if you use a MET-TEL3 inhibitor, which is STM2457, what we see that now this cell starts differentiating beautifully. So what we could see that if you block MET-TEL3 now, we actually could rescue the differentiation and phenotype in the MYCN overexpressed cells as well. And these results are good, right? Because in the neuroblastoma, the cells normally doesn't differentiate, and you see the tumor formation. But now if you're able to use a drug which can induce the differentiation, could have a tremendous therapeutic implication. And what surprising thing, what Ketan observed that, which we somehow sort of see in by chance or by luck, I will say, that when you treat these cells with the STM2457, we also see that these cells accumulate DNA damage marker, such as gamma H2X and phosphorylated RPA32. So we wanted to explore this using more therapeutic approach, that if we could combine now MET-TEL3 inhibition with the chemotherapeutic drug, which can normally cause the DNA damage, will that be sort of having a synergistic or additive effect? That is what exactly we tested. So if you add STM2457 in the MYCN amplified neuroblastoma cells, what you will see that the cells accumulate DNA damage. If you add doxorubicin, which is a chemotherapeutic drug, cells actually accumulate DNA damage as well, which is known, of course. But then now, if you combine STM2457 with the chemotherapeutic drug, you see that these cells drastically accumulate the DNA damage. And not only that, these cells are also quite compromised in the cell viability as well. But this is the data you see in vitro, right? You always want to see that does it happen in vivo, inside the body. Of course, you cannot do that in the human system, but we can, of course, do that in a mouse system, where we have utilized a mouse PDX. We have collaborated with the group in Cambridge who have access to MYCN amplified PDX model. So where mice were treated either with STM2457 alone, which is a MET-TEL3 inhibitor alone, or doxorubicin, which is a chemotherapeutic drug alone, or in combination with STM2457 and doxorubicin. And as you can see from the tumor growth, the purple line here is basically the combination of STM2457 along with the chemotherapeutic drug. And you can see very nicely that the combination has the much more greater effect on tumor reduction when you do these experiments in vivo in the mice. And not only that, what we also see that the mice body weight did not change much, and we sort of checked the other toxicity, like mice were eating well, drinking well. So we think that this didn't cause the much toxicity as well. So overall, what we concluded that oncogenic gene regulation by MYCN is controlled by M6 epitranscriptomic modification in the neuroblastoma cells. And we also think that MET-TEL3 inhibition in combination with the chemotherapeutic drugs could be a novel therapeutic approach for MYCN amplified high-risk neuroblastoma. So far, what we have discussed, this is the published data. So far, what we have discussed, that we see that the role of RNA modification in the telomere maintenance mechanism, which happens in the tera locus, it's a single locus. We also see that, or in the single RNA. We also see that the M6 RNA-mediated regulation of posterior HOX genes in the MYCN amplified neuroblastoma. But these analyses are limited to few genes, right? And seeing this important function in these cells, we want to explore the role of RNA modification in a more global way in a whole transcriptome in the neuroblastoma cells. And that is a part which I'm going to cover very briefly. So this is an ongoing project where we would like to map the RNA modification in the neuroblastoma using sort of more better method where we can sort of implement the RNA modification into neuroblastoma tumor samples. And these are the data which are unpublished data, and we have sort of utilized here a RNA profiling modification method called Epiplex, which I have discussed in the very beginning of my presentation. And I also mentioned to you that Epiplex allows us to profile the modification of the two modifications together, which is M6A and inosine, another RNA modification. So what we did as a sort of a pilot project, we have implemented Epiplex in 16 neuroblastoma samples from the different group, where you can recognize that this is a MYC amplified neuroblastoma tumors. These are the all positive neuroblastoma tumors, which we have also saw that it has an important role in the neuroblastoma tumor, in this tumor. And we also took as a control the low risk tumors. And as a kind of a validation, and we wanted to go back into the data and see that can it really sort of reproduce what we saw in our M6A dipsec. And if you remember that M6A dipsec, we repeatedly saw that the posterior Hox genes were M6A modified, right? And now you can see very nicely that Epiplex can do the same. And it's also very important to mention that we performed this dipsec before with around 5 microgram RNA, and now we did this experiment with the 200 nanogram RNA. So we have actually could reduce the RNA need by 25-fold. So, and this now allows us to perform such experiments in a larger number of the samples where the RNA amount is always the limitation. And if you now see that the M6A signal in this, which are detected by the Epiplex using this in these samples, you can see very nicely the MYC amplified tumors. They are sort of segregated in one place, whereas the low risk and all positive tumors, they are sort of localized, segregated in another place. And it is important to mention that you see that one of the low risk tumors are actually getting sort of clustered in the MYC group. And when we sort of look back into this sample and contacted our clinical collaborator, what is happening here? And we actually came to know that these samples are actually mislabeled, and it is actually a heterogeneous MYC amplified sample, which was mislabeled as a low risk sample. So actually looks like the Epiplex can unbiased to a sort of cluster MYC tumors. And I think I should also mention that we see that one of the red dots also are coming along with which is the all positive tumors coming together along with the MYC tumors. But this could be something which we don't know yet why it is happening. But this data comes from only 16 tumors, and we are sort of working on this project. And I guess more we do, we will know that is it like a more of the all positive tumors, do they say sort of a similar behavior with the MYC tumors or not? And this Epiplex comes along with the analysis pipeline, which is called EpiScout, and this can sort of identify the differential M6A methylation in the two groups of tumors. And here you see a sort of a snapshot of differential M6A methylation between MYC amplified tumors with the low risk, and you can see that this can efficiently detect the differential expression, differential M6A methylation, and across the samples. But I should also emphasize that this is the data which we had sort of just started analyzing it. So more we will learn from this data more. And with that, I have actually almost come towards the end of my presentation. I did mention a couple of times that Epiplex not only allows us to profile M6 RNA modification, but it also allows us to profile inosine modification as well. And as you can see that from the literature, we know that M6A modification shows a distribution pattern in the 3' UTR, and which we have also seen in our data repeatedly. But now, if you see that the inosine metagene plots, the inosine is located mainly in the coding sequences. So you could see that the two modifications, they show a very clear distribution pattern. And this is I have just shown the one tumor, but if you see across the tumor, we see that this repeated pattern of M6A and inosine distribution pattern. So we have not analyzed in very detail how the inosine modification is changing across the tumors, but this will be, I think, the exciting time for us to look into such data and learn more how inosine modification is actually affecting the neuroblastoma disease as well. So with that, I have almost come to my last slide of my presentation. So I will sort of end my presentation with the overall conclusion. and that I should sort of mention that using neuroblastoma as a disease model, I think I'm sort of, I have probably able to convince you that the RNA modification has a clear role in the oncogenic process. And I think we can appreciate that, that a deeper understanding of the RNA modification mechanism can help us to sort of design novel therapeutic strategies, such as, again, high-risk neuroblastoma. And I think advancing use the advanced epitranscriptomic profiling method will always have an advantage. So we got already started seeing that when we sort of ask for the clinicians that we need a 5 microgram RNA for M60 profiling, and now we can do it at the 200 to 300 nanogram, we could get much better response from them. And it's actually, well, we definitely think that such method can speed up the RNA modification profiling. And I also believe that the insights which we gain from this sort of data will uncover the unknown biology, which will not only be relevant from the cancer biology perspective, but it will be also relevant to the broader research field of RNA modification. So with that, I end the data slides, but I think I should take some time on this slide, because this is one of the most important slides as well. I would like to thank my collaborator who has been extremely helpful in last 4 or 5 years to get this work done along with me and help us get going. And this is my research group, current research group members and past research group members who has been sort of super enthusiastic and very hardworking to implement this project. And without their help, nothing could have been happened. And I should not also forget to sort of thank my funding agencies, which with that generous funding, it would not have been possible to implement such studies. I would like to specifically mention Burn Cancer Foundation, who have been quite generous to funding us in our project. And with that, I would like to end my presentation. I would like to thank you for listening. And if you are interested in the epitranscriptomic research, you are most welcome to approach us. We are happy to collaborate. And we will also like to, if you are interested in working with us, please do approach us. And this is our lab homepage. If you'd like to know, please go ahead and look into our homepage. And with that, I will hand over the mic to Andrew, who will explain in a couple of next slides about more on the Epiplex. And with that, I stop now and hand over the mic to Andrew. Thank you all for listening.
Thank you, Dr. Mondal. Very interesting talk and a really good story that highlights some of the interplay between RNA methylation and neuroblastoma oncogenesis.
Thank you.
So Epiplex is a product created by Alida. Alida was founded with the mission to make the epitranscriptome accessible. And towards this end, Epiplex allows us to start meeting this goal and offers the first end-to-end solution for RNA modification detection and analysis. To run this assay, you supply just the RNA, and this can be purified from a number of different sources. It can be from cells, fresh or frozen tissue, FFPE tissue, or blood. And to preserve the amount of RNA that you need to use for the assay, we suggest either 20 nanograms of poly-A enriched RNA or 250 nanograms of total RNA. The assay can be completed in a one-day workflow and includes all the reagents to go from RNA to sequencing-ready libraries. Under the hood, you create libraries that include a modification barcode that translates sequencing silent RNA modifications, such as M6A, into barcodes. This library can be sequenced on the short-read sequencer of your choice. It's compatible with sequencers from Illumina, Element Biosciences Complete Genomics. And Alida Biosciences has also developed an analysis pipeline known as Episcout, which I think Dr. Mondal briefly mentioned. This can be available either via cloud portal on DNA Nexus, or if you prefer to run it locally, you can download the container as Docker image and run on your own servers. So I mentioned briefly the proximity barcoding. So this is an enrichment assay. And so the RNA is fragmented, and the individual fragments are enriched on the surface of individual beads. Because this barcoding only happens at the surface, you can have a multiplexed reaction that's occurring within a single vessel. And so the fragments that contain M6A will only contain M6A barcodes. Ones that contain inosine will only contain inosine barcodes on it. And so you don't need to worry about splitting your sample into a whole bunch of different reaction vessels to probe different RNA modifications. Our current Epiplex kit currently probes both M6A and inosine, and soon we'll be including pseudouridine within our kits. But inherently, this technology is scalable to however many modifications we build specific binders for. I want to talk a little bit about the binders. We did approach this technology first by looking at antibodies. However, screening upwards of 20 different antibodies in some case for single modifications, we're unimpressed with the specificity. We got a lot of off-target binding with antibodies, and they took a lot of optimization. So we worked extensively to create engineered protein scaffolds that can enrich for modifications with very, very high sensitivity, but probably just as importantly, with very minimal off-target binding. Included within the Epiplex kit are spiking controls. These are essential to enable us to control for experimental variability. And so the spiking controls create a signal that's predictable, where we get a response that's proportional to the amount of modifications that's present in these spiking controls. So knowing the predictable response of these spiking controls, we can control for experimental variability from day to day. And finally, the software is optimized with the data type that we have, primarily the RNA modifications, RNA data sets, including our spiking controls, to identify sites of read enrichment, otherwise known as peaks within the data. And for some modifications as well, we can also find signatures that allow us to identify the sites of modification with single base resolution. The Epiplex kit is available as both 8 and 24 sample kits. As I mentioned previously, the workflow can be completed from isolated RNA to sequencing ready libraries within 1 working day. We suggest entering the assay with either 20 nanograms of poly A enriched or rRNA depleted sample, or 250 nanograms of RNA total. You can go in with less RNA with a slight degradation in the quality of the data. We support RNA isolated from a number of sample types. So of course, if you have highly pristine data with high RIN scores, isolated from tissue or cell lines, that works. We can also handle degraded RNA, even RNA from FFPE tissue. We support a number of short-read sequencers, and included within the assay is our EpiScout analysis software that can be run on the DNA Nexus portal or via Docker image. We found that this is really essential to be able to run the assay. So even if you don't have bioinformatics expertise on staff, you can still find biological insight in your data through the software that's available. If you're interested, we'd love to hear from you and discuss how we can support your biological questions. Please reach out to us, and thank you all for your attention. I'll now turn it back over to Catherine, who will moderate the question session.
Thank you, Andrew. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. We'll now start the Q&A portion of the webinar. The first question is, of the knockdown, to what level of reduction was the knockdown?
So could you repeat, Catherine, the question another time, please?
Of the knockdown, to what level of reduction was the knockdown?
Is that probably the question means that MET-TIL3 knockdown? I guess so, and I don't know what that wants to know. Is it like the level of we do see that the knockdown in the trunk neural crest cells, which is at the level of 50 to 60% in the neuroblastoma cells, we could almost knock down and 80 to 90% of the protein. But then I should probably highlight that these knockdowns are not the stable knockdowns, so where you sort of don't leave the knockdown condition forever, but we use the doxycycline to temporarily knock down, because we realize that if you sort of remove this protein and permanently from these cells doesn't survive. So I hope that this answered the question.
Thank you. Next question is, do you have an explanation why the MET3 inhibitor by itself has limited effect on tumor volume?
Yeah, so I mean, we do see that the only MET3 inhibition has a effect, but it's not that as drastic as the combination. But I think this is consistent with the data which we have in the in vitro. We did not see in the in vitro as well, like the cells were sort of not drastically inhibited for the proliferation. So there could be multiple reasons. Of course, the effect of the inhibition might not be optimal. So we are actually still collaborating with the groups who might have access to the better inhibitor. So I hope that with using the better inhibitor, we will be able to improve the efficacy of metal immunity alone. So we hope that the better metal-3 inhibitor will be able to sort of inhibit the cell proliferation and more than what we have used currently.
The next question we have is MYCN expression regulated consistently across branching lineages, or does it vary depending on its genomic locus and chromatin context?
So could you another time repeat the question, Catherine? I'm sorry.
Sure. Is MYCN expression regulated consistently across branching lineages, or does it vary depending on its genomic locus and chromatin context?
Yeah, so we do see that the MYCN expression was sort of changing in the different lineages. At least what we in our in vitro assay, what we see that the trunk neural crest cells had a very high expression of MYCN, and then it goes down. But then we also know that the MYCN expressed during the early development, and it also has expression in the not only in the sympathetic lineages but also in the brain as well. So to my knowledge, the MYCN expression happens during early embryonic development, but then MYC takes over in many situations. But in the sympathetic neuronal lineages, the MYCN has a very vital role during early differentiation process, and it should then go down. But probably what we see in the disease condition, such as neuroblastoma, that the MYCN is actually not going down because of the amplification of the MYCN oncogene, and that results in the tumor formation in such scenario. I hope that answered the question.
Thank you. We have another question. Have you tried looking into any other RNA modification other than M6A? Have you tried using mass spec to not just monitor M6A, but other RNA modifications while looking into the neuroblastoma? Yeah,
that's a very nice question. And we are actually currently sort of into it. So we have an ongoing project where we are trying to look for not only the M6A, but the other sort of modification. And one of the example is, of course, the inosine modification, which we are profiling with the Epiplex. But we are currently trying to look into the mass spec-based approaches, where we will like to know that if RNA modifications are differentially present in the different neuroblastoma groups. So that's an ongoing project, and unfortunately, we don't have much lead now, but hopefully some more leads coming on the inosine and soon.
Sure. Our next question. You indicate that loss of M6A function leads to loss of TERA foci and prevents TERA from localizing to telomeres. What is the underlying reason for this phenotype? Do we know the mechanism?
Yeah, so that's also a very nice question. So what we see that when you remove metal-3, we see that the TERA foci are lost, right? And the obvious question which someone will ask, will that leads to a decrease in the RNA level? So when we try to look many times the level of RNA, we saw a very minor change in the TERA RNA level. So what we believe that when you don't have the RNA modification, like M6 RNA modification, what happens, tera is actually failing to localize into the telomere, and that's probably happens because tera RNA is not able to form R-loop-like structure. So what are these R-loops? They are RNA-DNA hybrid structure. I have skipped that during my presentation because of the lack of time, but this hybrid structure can help you to help the RNA to sort of stick to the site of the transcription. And what we see that when you do not have the M6A modification, these R loops are destabilized. And that is probably the reason that the tera RNA is sort of not able to localize to the site of transcription, and then they are dispersed.
Thank you. We have a question for Dr. Price. In what research scenarios is concurrent detection of multiple RNA modifications most valuable?
So individually, the modifications are known to be regulators of gene expression. It's still an ongoing area of research how they interplay with one another. There are some examples. For example, I can think of, there's a recent paper showing maturation of macrophages where M6A is interplaying with inosine to control how they work together. But this is, an ongoing area of research, and we're hoping that tools such as Epiplex will enable more researchers to look into the interplay of modifications.
Thank you. Moving on, for Dr. Mondal, you mentioned that tera RNA contains both subtelomeric and telomeric repeat regions. Do you also detect M6A modifications within the telomeric repeat region, despite the absence of canonical DRAC motifs typically associated with M6A deposition?
Yeah, that's actually a very nice question as well. So we do see that the M6A modification is present inside the telomeric repeats RNA. So we use the mass spec-based approach and several molecular biology-based approach to show that these modifications can actually present in the UA triple G containing telomeric repeats. So even though they are sort of a non-canonical M6A modification sites, but what we think that even if it's a non-canonical sites, but we have a multiple of those in the RNA. So a lower level of M6A modification, but the multiple sites could have an effective function in the RNA function, RNA regulation. So we are still sort of trying to understand how this M6A is attracted into the non-canonical way in the tera RNA. And I hope that in the near future, I will have more insights on that to present here or in the other format as well.
Thanks. Could global effects of MET-L3 knockdown explain the tera foci loss? Did you test specificity with other approaches?
Yeah, so that's also a very nice question. That, I mean, once you knock down an enzyme like MET-L3, you will actually sort of disturb the function of many other gene, right? Because M6A is known to regulate many other gene function. But to sort of more convincingly see that this is specifically happens because of MET-L3 loss, what we did that after MET-L3 knockdown, we recruited by RNA CRISPR, specifically in the telomere or in the tera RNA, the MET-L3 as well. And what we could see that when you recruit specifically MET-L3 in the MET-L3 knockdown cells, we could recover the tera foci. So that suggests that the phenotype which we saw is not non-specific or not because of the secondary effect, but rather more specific effect of M6A loss in the tera because of MET-L3 knockdown.
Thank you. We have another question. Could tera RNA or its M6A modification status be developed into a biomarker for identifying alt+ tumors in patients?
Yeah, that's also a very nice question, and we are sort of working on the line as well. But of course, this comes with a bit challenge because like the tera RNA is a repeat RNA, right? So the mapping of these reads in the telomeric ends, it's always a challenge. But it's a very interesting question, and not only us, but the other group has shown that the tera level goes up in this all positive neuroblastoma tumor. So I guess it will be very, very interesting to see that if we could very quantitatively map this M6A modification in the tera and in the all positive versus all negative tumors, and definitely then I think that this could serve as a more like a prognostic marker for all positive neuroblastoma.
Thank you. Next question is, could global effects of MET-L3 knockdown explain the terafoci loss? Did you test specificity with other approaches?
Yeah, I think I answered to this question.
Did we do that already?
Yes.
Okay. You observed other one. You observed that MET-L3 and MYCN interact and co-localize across many genomic regions. Beyond their effects on posterior Hox gene expression, what additional functional impacts of this co-localization are known?
Yes, this is also an interesting question because we, of course, discussed during my presentation regarding HOX8 and HOX9 genes. And we also saw that in these genes, MET-L3 and MYCN sort of co-localize. But we also saw that this happens in globally in many other locations. What we believe that MYCN, and by its inherent nature to the binding to the DNA, it's sort of anchoring this MET-L3 enzyme to the different location, and thereby it gives the access to the RNA to the MET-L3 enzyme, and then the M6A modification can happen. And we have actually seen which I have not presented here, we do see that the genes which are co-bound are having a bit higher level of M6A modification compared to the rest. So I think that not only in the Hox locus, but this interaction is guiding the global M6A pattern as well in the MYCN neuroblastoma.
Great. Here's another question for you. How do you view MET-L3 inhibition as a therapeutic strategy for neuroblastoma? Given MET-L3's other roles in cells, is its inhibition likely to be safe?
Yeah, so that's also an interesting question. Whenever you use a drug, it comes with a safety concern, right? And I mean, we do know that the MET-L3 has a role in the other biological processes, but what we have seen until now in our mice experiment, that in the single drug alone or in combination with other drug, we have not seen any very drastic effect in the mice, no toxicity, which we have so far seen. And there are other reports from a group who has sort of discovered this drug. They have tested in the mice as well, and look into the hematopoietic lineages that they did not see much defect in the normal hematopoiesis as well. So I do believe that these drugs might have some toxicity, but it's not something which we should be very much worried about at the moment. But this is I'm not like a great expert here, but we always need to learn by doing. So I hope that we will learn soon more about this sort of results when we do more experiment.
Thank you. Our next question is, what is the reason for DNA damage observed following MET-L3 inhibition in MYCN-amplified neuroblastoma cells?
Yeah, that's also another nice question. So we actually saw that when you remove MET-L3 or you knock down MET-L3, we somehow surprisingly see that results. But then when you look into the literature, we found that the MET-L3 has been shown to important for the DNA repair process. And this repair happens by also the M6A modification. So I think that, like apart from their role in the telomere or Hox regulation, MET-L3 mediated M6A modification has also a role in the DNA damage response regulation, and which has been sort of explored by quite a few groups. So that could be the reason that MET-L3 knockdown results in the accumulation of DNA damage in these cells. And I think, sorry, that I should actually mention that this the peculiar nature of the MYCN tumors, because these cells are quite highly active for transcription. And when you have the high level of transcription, you always have a transcription replication collision, and these cells sort of tend to accumulate this DNA damage. And you need actually better repair process to take care of that. And if you sort of remove an enzyme which is required in this process, then you actually sort of make these cells more vulnerable. And that is why I think that the DNA damage or chemotherapy combination along with the MET-L3 inhibition could be quite effective in the MYCN neuroblastoma.
Thank you. That's all the time we have for today. We'd like to thank Dr. Mondal and Dr. Price and our sponsor, Alita Bio. If we didn't have time to get to your question, we'll try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.